And I'll finish with a virus that swept the world last year. This is pandemic influenza, this is swine flu, H1N1 influenza. And actually, just to go back, this incredibly beautiful glass model is by a sculptor called Luke Jerram. And one of the things the Wellcome Trust does is we work quite hard, not only on funding biomedical research, but we also fund public engagement with science. We have a gallery in London, and I would encourage any of you who are in London to come and visit it. We have an exhibition at the moment entitled High Society, which is about drug use. Our next exhibition is going to be on dirt. So we have quite interesting, by which I mean sort of, well, there's no dirt in here, but it's the sort of stuff that accumulates behind cupboards. And in fact we have that glass sculpture in the World Collection at the moment. But again, the methods that I described can now be used to analyse viral infections. And so it was possible to look at viral infections in very large numbers. And this shows something very important. So swine flu, when it came into the UK, this is the family tree of all of the swine flus that were sequenced. There were hundreds of them sequenced out of many, many thousands of infections. And what this starts showing you is it starts showing you that there were in fact multiple introductions to the UK. It wasn't one person coming in and bringing it in. It was introduced again and again and again and again. And what this analysis also shows is, because you can work out the family history and the length of genealogical trees, that in fact the first cases of swine flu in the United Kingdom occurred well before any of them were reported. So if one had closed the border and it was known beforehand that closing borders wasn't going to work for flu, it would have been too late, because it was in the country before any cases were identified. So you could just learn an extraordinary amount by sequencing viruses. And of course this is a world in which emerging infectious disease, emerging infectious disease is coming all the time. And of course Canada discovered that with the SARS outbreak a few years ago. This is just a map of the world with emerging infectious diseases all over it. So briefly, looking to the future, the challenges I think I've illustrated to you is linking genetic variation with phenotypic information. And they're both hard to do, but doing the phenotypes really well is extremely hard. And it's about mapping, sequencing, analysing different populations. And it's very much about scientists collaborating with each other. These are not studies that can be done by a single scientist working on their own in a laboratory. It really does require collaboration. But discovering the gene, as I've told you, is only the first thing. We have to work out how these genes are associated with both variation in health and disease. It involves working in other species. And of course, hardest of all in many ways, it involves the understanding of the interactions between genes and environment. And one of the studies that we funded in the UK, again in partnership with the Medical Research Council and with the UK Departments of Health, is called UK Biobank. And that's taken 500,000 volunteers aged between 40 and 69. And each of those volunteers has given DNA samples, and in fact I am a volunteer in that study, undergone all sorts of testing, exercises on bicycles, all sorts of things I don't normally do. And that we will all be followed until we disappear from this earth. And that will be a very powerful way of learning about genes and biology. But genes and genetic variation is also important for social reasons. And this is a project that I particularly like. It's an American project called the Innocence Project. And what they do is they use DNA information to actually release people from prison who've been wrongly convicted. And of course DNA is an extraordinarily powerful tool for determining genetic identity. So for forensic purposes it used to be thought of just as a barcode. But actually it contains a lot of content. You can work out from a DNA sample whether it comes from a male or from a female. You can work out what the hair colour of the individual is, whether they have red hair or not. If it's male and you have an uncommon surname, there's a reasonable chance you can work out your surname from your Y chromosome sequence. It's not a trivial probability of getting this right, and as we get more DNA information, it'll be better as that. So the question for society is, to what extent and how do we want to use this information? There are important questions. So, looking to the far distant future, will there be a world when everyone has their own genome sequence? Probably not in the foreseeable future. How will we think about deducing functional consequences? Will we have a society where we're comfortable about using DNA sequence information? To what extent is it going to influence medical practice? So I hope I've shown you over the last few minutes the extraordinary roller coaster that we're now riding. The 10th anniversary of Genome BC. Congratulations to Genome BC. I think that there's another 90 years of work at least, possibly another 300. Thank you for your attention.